If you carry type O positive blood, you are holding onto a genetic passport stamped by ancient migrations that predate written history, older than any empire that ever rose and fell, older than any border ever drawn on a map. For most of modern medicine's history, your blood type was treated as nothing more than a footnote, a letter, and a symbol printed on a hospital chart just to make sure a transfusion did not kill you. But that footnote is hiding one of the most dramatic survival stories in human evolution, and it starts with a body count. In the closing years of the 19th century, doctors across Europe and America who attempted blood transfusions were playing a horrifying game of chance with their patients' lives. Sometimes the procedure worked. Just as often, within minutes, a patient's blood would clump and curdle inside their own veins, sending them into violent shock and death right there on the operating table, with absolutely no way to predict beforehand which patient would survive and which one would not. Entire wards watched people convulse and die from a treatment that was supposed to save them. And by the late 1890s, some hospitals had quietly stopped offering transfusions altogether because the odds of killing the patient looked just as bad as the odds of letting them bleed out untreated. It took a single laboratory breakthrough in 1901 to finally explain why, and what it revealed did not just fix a medical mystery. It cracked open a genetic record stretching back tens of thousands of years, one that is still rewriting what scientists thought they knew about the letter sitting on your own medical chart right now. To understand how that laboratory breakthrough was even possible, you have to start with the man behind the microscope, an Austrian physician named Karl Landsteiner, working in Vienna in 1901. He had been staring at a problem that had killed patient after patient for decades, and nobody understood why the exact same medical procedure produced such wildly different outcomes. Landsteiner began mixing blood samples from different people under his microscope and noticed something nobody had documented before. Some combinations stayed smooth and liquid. Others clumped violently, the red cells clustering together like sediment. He had just discovered that human blood came in distinct, incompatible categories, which he labeled A, B, and O, with AB identified shortly after by his colleagues. It was a discovery so fundamental to modern medicine that it earned him the Nobel Prize in 1930, and it turned transfusion almost overnight from a terrifying gamble into a calculated, testable, and genuinely trustworthy procedure that doctors could finally rely on to save a life instead of ending one. But almost immediately, European researchers made an assumption that would take over a century to fully correct. Looking around at their own populations, where type A was extremely common, they concluded that type A must be the original, ancestral form of human blood, and that type O was some kind of later deviation, an oddity that appeared afterward. That assumption shaped medical thinking for generations. And it was built on a foundation that modern DNA sequencing has since turned upside down, though not in the direction that most popular retellings of this story like to claim. Here's where the science actually gets interesting, and where a lot of what gets repeated online about type O blood needs a correction. Your blood type is determined by a specific gene sitting on chromosome 9, a gene that acts like an instruction manual for building small sugar molecules, called antigens, onto the surface of your red blood cells. Every red blood cell in your body starts with a base structure called the H antigen. Think of the H antigen as bare scaffolding. If your genetic instructions tell your cells to add one additional sugar molecule onto that scaffolding, you become type A. If they add a slightly different sugar molecule, you become type B. If your genetic instructions are broken, if the gene responsible for adding anything at all has a mutation that disables it, nothing gets added, and your red blood cells stay in their original unmodified state. That unmodified state is type O. Today, roughly 37% of the global population carries some form of O-positive blood, making it one of the two most common blood types on Earth alongside A-positive. And that sheer prevalence is itself a clue to how successful this stripped-down molecular configuration has been across human history. In 2012 and 2013, a research team led by geneticist Laure Segrel at the University of Chicago published a landmark study in the Proceedings of the National Academy of Sciences showing that the underlying genetic system distinguishing A and B blood types is at least 20 million years old, inherited from a common ancestor shared by humans, chimpanzees, gorillas, orangutans, and several species of Old World monkeys. That is not a typo. 20 million years, long before anything resembling a human walked the earth. But here's the part that gets flattened in most popular science videos. The O allele itself is not some separate ancient lineage sitting alongside A and B since the beginning. Genetically speaking, O results from a deletion mutation, a small chunk of missing genetic code that disables the enzyme, responsible for building the A or B sugar structures. It arose independently multiple times throughout primate evolution as a loss of function, a genetic error that happened to stick around because, in specific environments, having a bare, unmodified blood cell turned out to be a survival advantage rather than a disadvantage. So the truer and honestly more fascinating story is not that O is the ancient original template untouched by time. It is that O represents a mutation that repeatedly succeeded over and over in different populations, because the environments those populations faced rewarded simplicity on the surface of a red blood cell. Nature did not preserve type O because it was first. Nature kept reselecting it because it kept winning. And nowhere is that victory more visible than in the story of how humans reached the Americas. Every strand of archaeological and genetic evidence confirms that the ancestors of Native American populations crossed from Siberia into Alaska across an ancient land bridge, sometime between roughly 20,000 and 15,000 years ago, during the tail end of the last ice age. This was an unbelievably harsh migration. Small isolated groups pushing through frozen, unforgiving terrain with no idea what lay on the other side. When modern researchers tested the blood type frequencies of indigenous populations across the Americas, the results were staggering. In some communities, nearly the entire population carries type O blood, and across Central and South America as a whole, type O positive blood is found in close to the entire population, some of the highest concentrations recorded anywhere on the planet. In genetics, this pattern has a name, a founder effect. It means the small original population that crossed into the Americas happened to carry an overwhelming amount of type O blood. And because they were so isolated from the rest of humanity for thousands of years afterward, that genetic signature never got diluted by outside populations. Every generation simply inherited what the founders carried. And when you trace that signature backward, it leads directly into ancient Asia, where type O remains extraordinarily common today. In the Philippines, roughly 45% of the population carries type O blood. In Vietnam, the number sits close to 42%. The genetic markers surrounding the O mutation in populations across the Americas match the markers found in East and Southeast Asia almost perfectly, giving researchers a genetic paper trail stretching back tens of thousands of years. But why would a population moving through brutal, disease-heavy environments end up so overwhelmingly stacked with one specific blood type? The answer has almost nothing to do with the cold, and almost everything to do with an invisible war that has been shaping human genetics for as long as our species has existed. Infectious disease. Malaria is one of the oldest and most relentless killers in human history, and it turns out your blood type is directly involved in whether you survive it. When the malaria parasite, Plasmodium falciparum, infects red blood cells, it relies on those cells sticking together, clumping into small clusters inside blood vessels in a process researchers call rosetting. This clustering is what causes the most severe, often fatal complications of malaria, including cerebral malaria. Type A and type B blood cells, with their extra surface sugars, cluster together far more easily. Type O cells, stripped down to that bare H antigen, resist this clustering. Clinical studies from malaria endemic regions across Africa and Southeast Asia have repeatedly found that individuals with type O blood are significantly less likely to develop the severe, life-threatening form of the disease. Across thousands of generations, in regions where malaria killed enormous portions of the population before they ever reached adulthood, that resistance was not a minor perk. It was the difference between a bloodline continuing or ending. Populations moving through tropical and subtropical zones on their way out of Africa and across Asia carried this advantage forward precisely because the people who didn't have it were disproportionately killed before they could pass on their genes. This is exactly the kind of pressure geneticists call natural selection acting in real time, visible not in some abstract textbook diagram, but in the living blood type maps of modern populations. In large parts of Africa, South Asia, and Southeast Asia, regions where malaria has been a dominant killer for thousands of years, type O blood remains unusually common today. And that concentration lines up almost perfectly with historical malaria maps drawn up by epidemiologists long before anyone connected the pattern back to a single gene on chromosome 9. But nature rarely hands out a gift without attaching a cost. And the case of type O blood is one of the clearest examples of that trade-off in all of human biology. Take cholera. In regions like the Ganges Delta in South Asia, where cholera outbreaks have devastated populations for centuries, researchers have found the opposite relationship. The cholera toxin produced by the bacterium Vibrio cholerae binds more efficiently to the surface of type O red blood cells than to A or B cells, and clinical data from severe outbreaks consistently shows that people with type O blood suffer dramatically more severe dehydration and higher mortality once infected. This actually explains a real geographic pattern. Type O blood is measurably less common in parts of India and Bangladesh, where cholera has historically been most devastating, almost as if the disease itself edited the local gene pool over centuries, quietly filtering out the trait that made people more vulnerable. Then there's the stomach. People with type O blood carry a meaningfully higher lifetime risk of developing peptic ulcers, largely because a corkscrew-shaped bacterium called Helicobacter pylori binds far more effectively to specific molecules present on type O cells lining the stomach and digestive tract. In populations across East Asia, where H. pylori infection rates are extremely high, researchers closely track the connection between type O blood and elevated rates of gastric complications. And if you want a smaller, stranger example of this same pattern, consider mosquitoes. In controlled experiments where volunteers exposed their arms to hungry mosquitoes in enclosed testing boxes, insects landed on and bit people with type O blood at roughly double the rate of people with type A blood, apparently picking up on some chemical signal in the skin linked to blood type. None of this makes type O blood good or bad. It makes it a very specific evolutionary bargain, a set of trade-offs forged by exactly which diseases your ancestors were fighting and exactly which environment they were fighting them in. Which brings us back to a case from Bombay in 1952, and a mystery that blood typing alone could not solve. A patient was admitted to a hospital there and tested as type O, but when doctors gave them standard type O blood for a needed transfusion, the patient's body violently rejected it. When researchers dug deeper into that case, they discovered something that reshaped how doctors think about the letter O entirely. Remember that every red blood cell, regardless of blood type, is supposed to carry that basic H antigen scaffolding, the raw foundation that type A and B cells then build additional sugars on top of. Type O blood was assumed to simply be cells that stopped at that foundational stage. But this patient's cells did not even have the foundation. There was no H antigen present at all. Under a standard blood test, his blood looked exactly like type O because it had no A or B markers to detect. But biologically, it was something else entirely, a vanishingly rare condition that came to be known as the Bombay phenotype. People with this condition can only safely receive blood from other Bombay phenotype donors because their immune system will violently reject blood from literally any other source, including standard type O. Globally, this condition appears in roughly 1 in a million people. But on the Indian subcontinent specifically, it shows up far more frequently, in roughly 1 out of every 10,000 people, a localized genetic quirk embedded deep in South Asian population history that standard blood typing has no vocabulary to describe. The Bombay phenotype remains one of the clearest reminders in all of modern hematology that even a category as seemingly simple as type O contains hidden layers that took researchers decades and a genuine medical emergency to even discover. The most direct evidence for how and when all of this genetic sorting actually happened has come from an entirely new frontier of science, reading blood type genetics directly out of ancient bones. For most of the 20th century, researchers could only study blood type evolution indirectly, by comparing living populations and inferring what must have happened in the deep past. That changed with the rise of ancient DNA sequencing. In a major study whose findings became widely reported starting in 2024, researchers analyzed the genomes of 22 Homo sapiens individuals who lived between roughly 46,000 and 16,500 years ago, alongside 14 Neanderthal individuals and 1 remarkable individual descended from both a Neanderthal parent and a Denisovan parent, dating back close to 100,000 years. What they found was striking. Neanderthal blood type genetics barely changed across 80,000 years of their existence, a sign of small, isolated inbred populations with very little genetic flexibility, a pattern some researchers believe contributed to their eventual extinction. Early modern humans showed the opposite pattern entirely, with blood type variants appearing and shifting rapidly as populations moved into new environments and encountered new diseases. One especially compelling piece of this research points to the Persian Plateau, a region between the Zagros Mountains and modern-day Iran, where archaeological evidence suggests early Homo sapiens groups paused during their migration out of Africa for something like 15,000 years. Researchers now believe this extended stopover functioned as a kind of genetic incubator, a long enough window for new blood type variants, the ones found almost exclusively outside Africa today, to emerge and stabilize before those populations continued spreading across Europe and Asia. Every population alive today carrying variants first forged during that 15,000-year layover is walking around with a direct genetic echo of that ancient pause. That picture of accelerating genetic change did not stop with the Persian Plateau. In 2026, a massive ancient DNA study led by researchers including David Reich and Mohammed Akbari, drawing on close to 16,000 ancient human genomes gathered from archaeological sites around the world, found that human evolution has actually sped up over just the last 10,000 years, a blink of an eye by evolutionary standards. The study identified hundreds of genes that show clear signs of natural selection during that window, tied to immunity, skin pigmentation, diet, and behavior, all responding to the explosive changes that came with farming, permanent settlements, and denser populations living in closer contact with each other and with domesticated animals. Blood type genetics sits squarely inside that accelerated window. The same rise of agriculture in crowded early cities that reshaped immune genes across the globe was also the backdrop against which local blood type frequencies kept shifting, tipping toward whichever configuration gave a population the best odds against the specific pathogens circulating in their exact region at that exact moment in history. That same drive to spread, adapt, and survive shows up again in one of the most remarkable maritime achievements in human history. Roughly 3,000 to 5,000 years ago, seafaring populations, originating from what is now Taiwan, began an extraordinary expansion across the Pacific Ocean using outrigger canoes, eventually reaching and settling islands as far apart as Hawaii, New Zealand, and Easter Island. When modern researchers test the blood type frequencies of Polynesian populations descended from these voyagers, they find sharply elevated levels of type O blood, echoing the same founder effect pattern seen in the Americas. Two of the most geographically isolated population groups on the planet, Indigenous Americans, separated by the Bering Land Bridge, and Polynesians, separated by thousands of miles of open ocean, ended up carrying strikingly similar concentrations of the exact same ancient blood signature, despite having no contact with each other for millennia. The pattern is not coincidence. It is the fingerprint of small founding populations, harsh selective environments, and centuries of isolation, repeating itself independently on opposite sides of the globe. Beyond infectious disease, type O blood carries measurable consequences for your cardiovascular system that have nothing to do with lifestyle or diet. People with type O blood naturally produce lower levels of two specific clotting proteins, factor VIII and von Willebrand factor, compared to people with type A, B, or AB blood. Lower levels of these clotting factors translate into a meaningfully reduced risk of dangerous blood clots, the kind responsible for deep vein thrombosis, certain strokes, and specific types of heart attacks. This is not something you can train or diet your way into. It is a direct expression of the same genetic architecture that determines your blood type. The trade-off, unsurprisingly, is that type O carriers also tend to experience slightly longer bleeding times when they are cut or injured, since the same clotting proteins that raise clot risk in other blood types also help wounds seal faster. The same clotting biology resurfaced in an unexpected way during the global COVID-19 pandemic, which functioned as an enormous, unintentional real-world experiment in blood type immunology, with data available from millions of patients across dozens of countries. Multiple studies published during and after the pandemic found that people with type O blood, and separately people who are Rh negative, showed somewhat lower rates of testing positive for the virus, and appeared to experience fewer of the most severe vascular complications, including dangerous clotting events linked to elevated von Willebrand factor levels in patients with other blood types. Researchers were careful to note this was a correlation observed at population scale rather than a guarantee for any individual, but the pattern lined up closely with decades of existing research on how type O blood interacts with clotting and inflammation. It also lined up with something hospitals have known for a very long time outside of any pandemic. Type O negative blood, lacking A, B, and Rh markers entirely, is the one type that can be given to almost any patient in an emergency without waiting for lab confirmation, which is why it is kept on hand for trauma cases, for premature and immune-deficient infants, and for any situation where there is no time left to run a full blood test before a transfusion has to begin. Modern science has also completely exploded how many distinct blood group systems actually exist inside the human body. Most people grow up believing blood type begins and ends with A, B, AB, O, and the Rh positive or negative designation. In reality, the International Society of Blood Transfusion recognized 47 distinct human blood group systems by October of 2024. Following the formal identification of the MAL system by researchers at NHS Blood and Transplant and the University of Bristol, solving a genetic mystery around a rare antigen called ANWJ that had puzzled scientists for roughly 50 years, ever since it was first observed in patients whose blood simply would not behave the way any known system predicted. Within less than a year, in 2025, a 48th system called PIAXZ was identified through a newly discovered gene mutation affecting how certain proteins anchor themselves to the surface of red blood cells. Each of these systems represents its own distinct set of surface markers, its own evolutionary history, and in rare cases, its own life or death clinical significance for patients who need precisely matched blood and cannot receive standard transfusions safely, including patients like that Bombay hospital case decades earlier, whose survival depended entirely on doctors recognizing a system standard testing had never been built to detect. The science of blood typing, in other words, is nowhere near finished. It is accelerating, with new discoveries reshaping the picture almost every year. That acceleration is also pushing researchers towards something that sounds like science fiction, but is already an active area of laboratory research, converting other blood types directly into type O. Because type O negative blood functions as the universal emergency donor type, and type O blood in general is constantly in short supply relative to demand in hospitals worldwide, researchers have spent years investigating specific enzymes, glycoside hydrolases, capable of clipping away the extra sugar molecules that distinguish type A and type B blood cells, effectively stripping them back down to the bare H antigen foundation that defines type O. If this enzymatic conversion process is ever perfected at a clinical scale, it could dramatically ease the chronic global shortage of universal donor blood, allowing hospitals to convert more common blood types into the one that emergency rooms depend on most. There is something almost poetic in that goal. With every advance in gene editing, sequencing technology, and laboratory biochemistry, the destination researchers are engineering their way toward is the same stripped-down, unmodified cellular structure that a random genetic deletion produced over and over in different populations across tens of thousands of years of human history. So what does all of this actually mean if you are one of the more than 1/3 of the global population walking around with type O positive blood today? It means the letter and symbol on your medical chart is not a neutral administrative detail. It is a compressed record of a specific evolutionary bargain your ancestors made. Whether they crossed a frozen land bridge into an unknown continent, paddled outrigger canoes across the widest ocean on Earth, or survived generations of malaria in the river valleys of ancient Asia, it means your lower clotting risk, your particular vulnerabilities to cholera and stomach ulcers, and your position at the center of nearly every modern emergency room's blood supply are not random. They are the accumulated result of an immune system inherited fully formed, not learned during your own lifetime, but passed down intact from ancestors who survived pressures most of us will never have to imagine directly. Every new study, every ancient genome sequenced, every new blood group system identified, adds one more page to a story written in molecular detail, long before anyone thought to look for it. Scientists are still uncovering how deep that story goes, and what other advantages and vulnerabilities remain hidden inside the DNA that determines something as seemingly simple as your blood type. As sequencing technology keeps getting cheaper and ancient genomes keep getting recovered from sites all over the world, researchers expect the map of blood type history to keep filling in, revealing migrations, bottlenecks, and disease pressures that no written record could ever have preserved. Your blood is, in a very real sense, a document, one written not in ink, but in molecules, copied and passed forward with only occasional edits across an unbroken chain of survivors, stretching back long before anyone thought to write anything down at all, long before that first desperate transfusion attempt in a 19th century operating theater, and long before Landsteiner ever looked through a microscope, and finally gave that ancient inheritance a name. If this changed how you look at the letters on your own medical chart, drop a comment and let me know your blood type and where in the world your family history traces back to. Thank you for watching, and I will see you in the next one.